Right, now we're going to talk about the different concentration of solution and how it affects the cells. So we're going to have three different type of concentration. We have the hypotonic solution, the isotonic solution, and the hypotonic solution. I'm pretty sure you have heard of this term before, isotonic, where we find it in mostly in isotonic drinks. We have the 100 plus and we have Gertrude, if I'm not mistaken, that's the name. Alright, so what happened here, for the hypertonic solution, mostly we talk about anything that has a concentration below 7%. Isotonic is somewhere between 8.5% to 7.5%. Right, and then we have the concentration of isotonic where it falls between 7.5% to somewhere around 11%, and hypotonic it's more than 12%. So in terms of mole per dm cube, the hypotonic can go somewhere below 1 mole per dm cube. For isotonic, somewhere around 1 mole per dm cube. And for hypotonic, it's above 2.5 mole per dm cube. Right? So these are the concentration for each type of solution. It varies from books to books. Even your textbook have their own definition. Why does this happen? Is because for this type of concentration, they differ from molecules to molecules, differ from ions to ions. So when we talk about this hyper-iso-hypo, for examination purpose, we're going to talk about only three substances. We're going to talk about salt, which is your NaCl. We're going to talk about the glucose. And we're going to talk about the fructose. And sometimes they put in sucrose just to compare between the different types. Alright, so let's start with the hypotonic solution. For our blood cells, our model cells are usually the red blood cells. So what happened? The red blood cell, they are biconcave in nature. You're going to learn this later in chapter 9. So in hypertonic, what happened is that since the concentration is lower, water moves out of cell. So please memorize this. The water, they move out of cells. So what happened to your cell? Your cell will start to shrink and they undergo the process known as crenation. Right? So here water flow out of cells, so net flow out of cell. And then we have the isotonic where water movement inside and outside of the cells is the same. So here water moves in, here water moves out. What happened here? Here we have net flow 0. And then we go to the hypotonic, where the hypotonic, we have a higher concentration of water